everyone, we are from Group 4. Here are my members' names. We are going to present topics on microarray and expression sequence tags. First thing first, microarray. Do you know what is it? DNA microarray helps us to determine whether individual DNA contain mutated gene. To use microarray, the most important thing that we need to have is the chip. The chip consists of a small glass plate encased in plastics. Next, we move on to the techniques of using microarray. First, we need to obtain DNA samples from patient's blood as well as control sample. Second, we will denature the DNA in the samples so that we can separate two complementary DNA into single-stranded molecules. Then, cut long strands of DNA into smaller and more manageable fragments. After that, label each fragment by attaching a fluorescent dye. For the individual DNA, we will use the green dye. For control DNA, we use the red dye. Next, send them both to chip. It is to allow the dye to hybridize or bind to synthetic DNA on the chip. Finally, we move on to the result and analysis of the DNA samples. If the DNA is normal, red and green dye will bind to sequence with no mutation, while if mutation occurs, individual's DNA will bind to sequence that represents mutated DNA on the chip. Here comes the applications of microarray on cancer research. They are drug discovery, identify cancer biomarkers, single nucleotide polymorphisms, classification of tumors, and identify gene target of tumor suppressor. This is the example of DNA microarray in medical microbiology, which is being used to identify Bacillus anthracis based on the rRNA intergenic transcript spaces region. Next, we move on to the expression sequence tags. Express sequence tags, or EST, known as short, unedited, randomly selected sequence reads derived from cDNA libraries. EST are short sequence reads which typically within the range of 100 to 700 base pair. An EST is a sequence tag site STS derived from cDNA. It is generated by sequencing a single segment of random clones from a cDNA library. What EST are for? EST can be directly used for gene discovery, proteome analysis, single nucleotide polymorphism characterization, alternative splicing, and gene structure identification. However, EST are error-prone and require multi-step pre-processing followed by clustering, assembly, database matching, and functional annotation to yield transcriptome information. Most EST have been isolated from humans. A large number of EST have been isolated from model organisms. To date, over 45 million EST have been generated from over 1,400 different species of eukaryotes. Next, we proceed to the techniques on how to use EST. First, raw EST sequence from chromatograms and database are checked for vector contamination, low complexity and repeat regions, which are excised or masked. Low quality, singleton, and very short sequence are discarded. Then, high quality ESTs are clustered and assembled to generate consensus sequences. Third, DNA database similarity search are carried out to assign, identify homologous, and assign possible function. The fourth step is the conceptual translation of consensus EST sequences to produce putative peptides. The fifth step is the performing protein database similarity search to assign putative functions. Then, the analysis is extended to functional annotation. And finally, visualize and interpret the result. Genetic markers and gene therapy are the examples that have been used in EST. That's all from our group. Hope you enjoy. Thank you.
Created using Powtoon.